This RPA is about food tests, about testing to see different food types if they contain sugar or glucose, I should say, starch, protein or fats, lipids. So I've got three things here which are going to give us a positive test. We'll start with protein. Now the test for this is biuret or burette reagent. A reagent means a chemical that is going to change colour in some way to indicate a reaction has happened, or indicate, in this case, the presence of protein. So biuret is pale bluish for protein. Notice it's got the corrosive symbol on the back, so if we get it on our hands, of course we'd wash it off. I could wear gloves. I am wearing goggles for safety, but we get the idea. Now, rather than using the little dropper, which will take me a long time, I'm just going to pour a bit in carefully. There we go. Now at first it looks sort of the same palish blue colour, but I'm going to mix it. I hold it at the top of the tube and I just gently shake it backwards and forwards, agitate it, and you can see now if I hold it against something white, you can clearly see that purple colour is different to the original blue colour. So that is a positive reaction. The milk powder contains protein, which is what we'd expect. Next, this is actually a bit of cereal, so this is going to be the test for starch. This is iodine. Again, it's got the warning hazard symbol on it, which it means it's a general sort of hazard. It can be used quite safely as an antiseptic. So some of you may have even had this on your skin if you ever had to have a hospital. Things like surgery would use it. So pop a bit in there. Now sometimes you might do this on a little separate slide because it can be a bit difficult to see the colour change in here, but you get the idea. The colour change you're looking for is it goes blue or black. Now again, if I hold this sort of tilting it up, you can see that the flakes inside have actually gone black. Okay, depending on what you put in, sometimes it looks a bit bluish. Sometimes the colour of the food itself can alter your perception of the test itself. But that is a positive test for starch. We've got that nice dark bluey black colour. Now the test for sugar, sometimes people say, more correctly we should actually say that this is the test for glucose, because there are different types of sugars and not all of them give you a positive result. This one is another blue colour, Benedict's reagent. Again, it's going to change colour for us if there is glucose present. However, if we put our stuff in, just make sure we've got enough in there. This is biscuit, so it should have some in there. Even if we give it a little shake up, nothing's going to happen, because in order for this reaction to occur, this must be heated up. So what I'm going to do is I'm going to put that into a water bath, basically some hot water. It needs to be about 80 degrees. I'm going to let that sit there for a couple of minutes and see if we get a colour change. So after a couple of minutes of heating, here's my positive test for glucose, and depending on how much glucose is in there, it will actually change colour. It's kind of a greeny, almost starting to go yellow at the moment, and because I know that the sample I used had plenty of glucose in, it will actually start to go orangey, if not red. So I'm just going to leave that running for a second, and I'll speed the video up to make you so you don't have to waste so much time waiting for it. Okay, so that's going now a much nicer, looks quite a mucky sort of orange colour, but it's telling us there's plenty of glucose in there. So that's now been in there about 5 minutes, and we've got this almost red colour really when you look at it. It's important to recognise at this point as well, what's forming in here is something called precipitate, which means little particles that are so small that they stay sort of suspended in this liquid. If we leave it long enough, they'll all settle to the bottom like mud going down in water, but they're so small that they tend to just stay there, particularly while it's warm, and that they stay suspended in that liquid. But if we leave it long enough, the precipitate will settle to the bottom. We could actually take it out and dry it out and weigh how much precipitate there is. That would give us a way to judge how much glucose is in that food. The simpler way to do it is just look at the colour. So anywhere between that green, yellow, orange and indeed red will give you an indication of how much there is in there.